Welcome, students, to today's lecture on embryology. Today, we will be delving into the fascinating world of sexual development and the intricate processes that contribute to the formation of the reproductive system. This is a complex and multidisciplinary field, encompassing genetics, molecular biology, endocrinology, and developmental biology. By the end of this lecture, you will have a thorough understanding of gametogenesis, gonad development, sex determination, and the formation of the external genitalia. In the process of gametogenesis, germ cells undergo meiosis to form distinct male and female gametes. This process occurs in the testes and ovaries and is regulated by hormones such as FSH and LH. During gonad development, intricate genetic and molecular mechanisms determine whether embryos develop testes or ovaries. Sex-determining genes, like SRY, initiate and guide this process. The presence of the Y chromosome with SRY leads to testes development, while its absence leads to ovaries. These processes involve complex interplay between genetics and signaling pathways, ultimately determining sexual differentiation. Sex determination is a complex process involving the integration of genetic, chromosomal, and hormonal influences. While the presence of the Y chromosome is considered the primary determinant of male development, there are various mechanisms by which male or female phenotypes can arise. These mechanisms include the presence or absence of specific sex chromosomes, the regulation of sex-determining genes, and the presence or absence of certain hormones during critical developmental windows. The SRY gene, located on the Y chromosome, is of utmost importance in male sex determination. This gene encodes a transcription factor that orchestrates the early events of testicular development. The SRY protein activates a cascade of molecular events, leading to the differentiation of supporting cell lineages and the subsequent production of testosterone. Understanding the molecular mechanisms underlying the function and significance of the SRY gene provides valuable insights into male sexual development. The formation of the Mullerian ducts is a step-by-step process crucial for the development of the female reproductive system. This intricate process involves the expression of specific signaling molecules, such as anti-Mullerian hormone, AMH, and intricate cellular interactions. The Mullerian ducts give rise to the fallopian tubes, uterus, cervix, and upper vagina. The precise coordination of molecular signals and cellular interactions is essential for the proper development of these structures. In contrast to the formation of the female reproductive system, the male reproductive system develops from the Wolffian ducts. This development is driven by hormonal signaling, primarily mediated by testosterone and dihydrotestosterone, which are secreted by the developing testes. Hormones act on the Wolffian ducts, causing their differentiation into the epididymis, vas deferens, and seminal vesicles. The interplay between hormones and cellular interactions intricately guides the formation of the male reproductive structures. Sex hormone regulation is a complex process that involves intricate feedback loops and complex interactions. Testosterone and estrogen, the primary male and female sex hormones, respectively, play critical roles in regulating genital development and sexual differentiation. These hormones influence the development of the reproductive structures, secondary sexual characteristics, and overall sexual phenotype. The delicate balance and interplay of these hormones are crucial for normal sexual development to occur. The formation of external genitalia is a complex process involving signaling molecules and tissue interactions. Structures like the penis, clitoris, labia, and scrotum develop from common precursor tissues through differential growth and morphogenetic events, regulated tightly to ensure proper formation. The genital tubercle is a critical structure that gives rise to the external genitalia in both males and females. Its development is modulated by various molecular and cellular factors, including the differential expression of homeobox genes and hormone signaling. Disruptions in the development of the genital tubercle can lead to anomalies such as hypospadias or epispadias. Understanding the intricate developmental processes and factors involved in tubercle formation provides insights into the etiology of congenital abnormalities. The formation of the final reproductive organs involves highly coordinated processes of fusion and separation. Structures such as the urogenital folds, labioscrotal swellings, and urogenital sinus undergo specific cellular and tissue interactions to create the distinct male and female reproductive organs. Precise regulation of genes, hormones, and signaling molecules ensures the proper fusion and septation events crucial for the establishment of functional and separate reproductive systems. The development of variations in genital development can result from genetic and developmental abnormalities. Intersex conditions encompass a range of disorders that affect sexual differentiation, including chromosomal abnormalities, such as Turner syndrome or Klinefelter syndrome, and hormonal disorders, such as congenital adrenal hyperplasia. These conditions have significant impacts on sexual development and can present challenges in diagnosis, treatment, and personal identity. Sexual differentiation disorders encompass a wide range of genetic and hormonal abnormalities that disrupt normal sexual development. Conditions such as androgen insensitivity syndrome or congenital adrenal hyperplasia result in atypical formation of the reproductive system and can have varying manifestations. Understanding the underlying causes and mechanisms of these disorders is crucial for proper diagnosis and management. Sexual development is a complex process involving genetics, molecules, and hormones. Understanding it helps diagnose and support individuals with sexual differentiation disorders. Proper regulation ensures the development of a healthy reproductive system.